All right, welcome everyone, and a special welcome to our guest speakers, whom we are extremely grateful for taking the time out of their day to come and chat with us about UniMelb research on alternative protein. I'd like to first begin with an acknowledgement of country. I'd like to show my respect and acknowledge the traditional custodians of the land of elders, past and present, on which this meeting takes place in Narm, also known as Melbourne at this time. So a little bit of an introduction. My name is Marion, a Master of Biotechnology student at the University of Melbourne, and I will be your MC today alongside Dawson, a final year of Bachelor of Commerce student at the University of Melbourne. Say hi, Dawson. Anyways, the Alternative Protein Initiative is a student-led group that aims to generate interest and excitement in the field at our university and the public. Our long-term vision is to build a healthy, sustainable, and just food system by creating a vibrant and robust community for alternative protein research, innovation, and entrepreneurship, starting from the University of Melbourne. So if that sounds interesting to you, make sure to follow us on our Facebook page, where we regularly post about our upcoming events, and send us a message if you're interested in getting involved in the initiative as well. So I know that this event has been shared quite extensively to many groups like the Biotech Club, the Biological Society, Effective Altruism Club, Vegan Club, Tissue Engineering Group as well. So I welcome everyone, and it's heartwarming to see us all coming together from different interests and fields to think about alternative proteins and how we can solve this pending protein problem. So we want to welcome you to today's event, the Future Food for Thought, a talk and panel discussion on research projects and opportunities in alt protein at the University of Melbourne. And we have four wonderful speakers. So we got Dr. Minha, Dr. Gregory Martin, Kieran Watkins, and Brody Peace, talking about the Future Food Initiative and their respective projects. After the talk, we will dive into Q&A and panel discussions on the research experiences and the opportunities with the Future Food Hallmark Research Initiative. And we'll be collecting questions on Slido throughout the talk, and we will use it to supplement the panel discussions. I've just posted the Slido link in the chats. I think Marcella, if you can post it again, that would be great. We'll also be using this link throughout the talk to ask questions to you so that we can figure out what events to organize in the future. We have a new question up almost after every speaker, so just keep checking the poll between the talks to fill it out. It'd be very much appreciated. Finally, I'd like to ask for participants to keep their microphones off during the presentation. This session is recorded, and the recording will be made available later on. We'll post the details of this on the event page. With that out of the way, I'd like to introduce you to the first speaker, Dr. Minha. So Dr. Minha is the academic convener of the Future Food Hallmark Research Initiative at the University of Melbourne. He is a research fellow researching proteins and associated nutrients in different foods, their sensory and nutritional qualities, and how different food technologies affect them. He applies the fundamental knowledge on conventional proteins in current research on alternative proteins, including plant proteins and cell-based meat. Today, he is here to introduce us to the Future Food Initiative. He has to leave a little bit early, so if you have any burning questions to ask him, please put it on Slido so we can prioritize these questions to him for the panel discussions. Anyways, here is Dr. Minha.
Thanks, Marianne. That's a very big welcome. I just better than expected. Thanks everyone for joining. I'm not going to talk a lot about my research. It's really just a quick overview of the Future Food Hallmark Research Initiative at Melbourne Uni, how the initiative came about and what we're doing exactly, and how you can be involved. With that, I'll share the screen of just a few slides I put together. Okay, so that's the link to the website of the Future Food Hallmark. There, if you're interested to look into our research projects a bit more, and I've got my contact details there. Now, the second contact details, the second email address is a general inquiries to all hallmarks. So there are many hallmarks at Melbourne Uni. This particular one focus on future food, in specifically proteins and future protein production. So pretty much our rationale of how we came about with this research initiative is really, now we know a lot about conventional proteins, and that's mainly meat and dairy and eggs and so animal-based proteins. And so if you look on the second column there, our knowledge has spent pretty much a huge over the last few decades, get a lot of knowledge, a lot of understandings of how these proteins come about, their functionalities, how to maximize the yield, how nutritious they are, and how sustainable or not sustainable they are, what health benefits they might have, what kind of industry applicable technologies we can apply to these proteins production systems and maximize all of those benefits, minimize all of the downfall, so to speak, of these protein sources. Now, one of the key area of the mainstream protein production is obviously for marketers and for the industry is the loss of a big group of the consumers, and that's the vegetarian, the vegan consumers. And for whatever reasons, they might be switching to the alternative protein sources. And so, over the last probably decade or so, the alternative protein research really came about, and it is surging with a vengeance at the moment. And so our rationale is, well, for the industry, certainly here in Australia, to adopt those technologies, those new protein sources, we really need a lot more research in that area. And so a few of our protein sources of focus in our Future Food Hallmark research initiative is the plant proteins and the non-animal analog. So, the meat proteins, the seafood proteins, even eggs and milk proteins, but not derived from animal growing. So the focus really is to integrate knowledge that we already have in the conventional protein productions, apply that in the alternative proteins, and stimulate a lot more research and industry collaboration in that area. So just very quick overview that we know that protein consumption is only growing and growing exponentially across the whole world. Now, with GDPs growing in every single country, there's a direct correlation between protein consumption and GDP growth. If you look at this graph here, for example, it's showing where the GDP is. I can't remember which year now, that's quite old now, 2013, versus the protein consumption. And pretty much what it shows is the higher the GDP, the more protein consumption per capita in those countries. A breakdown of the FAO numbers to see where the proteins are coming from in each of these countries. Pretty much the conclusion is that each country is slightly unique, but the main theme of the similar theme is that it's coming from a whole wide range of sources: animals, plants, offals, fruit, vegetables, and so on. And so it depends on where the industry is targeting. Then they really need to, when I say where, I mean which country for export purpose, then that's where they really need to focus on the source of the protein that's familiar to the consumers in those countries. Now, zooming into the Australian setting, what we see is that Australia, we get a lot of our protein, mostly from our conventional sources, meat and dairy. We also get a lot of them from carbohydrate source kind of food. So we don't tend to link them to proteins, but things like bread and pasta and confectionaries, even there's proteins in there as well. How healthy are they? I'm pretty sure we all know what kind of food is good for us. Now, just lastly, before I go into the Future Food Hallmark Research Initiative, this is the most recent numbers or figures for the vegetarian and the vegan consumer segments in each of these countries. And as we see, there's in countries like India, there's an enormous population of vegan and vegetarian. In Australia, we have a lot less, but that number is growing. And the last time I heard, Australia is actually one of the fastest growing markets for vegetarian and vegan consumers. So that proportion is changing fast in Australia. So that brings me to the Future Food Hallmark Research Initiative. This is a multidisciplinary research initiative that's pretty much pulling together 6 different faculties. So the core researchers are coming, the host faculty is veterinary and agricultural sciences, and we've got the faculty, so engineer at the School of Chemical Engineering mostly, faculty of arts, of sciences, business and economics, and also medicine, dentistry and health sciences. We also got external collaborators from the CSIRO and also from private research institutes and companies as well. So we're coming from different angles, different perspectives, trying to solve the main question, which is alternative protein. So the pretty much the foci of our initiative is on sustainability, is on community, personal and health and well-being, to promote industry and university collaboration, and also to provide a competitive edge for the Australian and the Victorian agribusiness and food sector. So that's our knowledge and collaboration. So there are 5 main themes of research in our initiative. The first one is to understand the drivers behind alternative proteins. That's coming from the community, from industry sectors, from government sectors. So trying to understand what's influencing that market growth. The second one is understanding from the consumer's perspective, what are their attitudes, what are their mindsets, why are they choosing or not choosing alternative protein, how do we segment the consumer market into different segments, and how to best position a particular product towards that segment of the consumers. The third theme is the health and well-being. So this is where we focus a lot on nutrition, on which alternative protein sources are best for which age group, for which gender, for pretty much different consumer segments. The third one is looking at various production systems. So that's plant proteins. We know a lot about it, but we are now doing a lot more research in fermentation, in cell-based meat, and we're now even talking about dairy production using fermentation techniques. And the final research theme is proof of concept. So now that we've got all of these alternative sources of proteins, what kind of products can we make, and how do we bring that kind of proof of concept product into sensory? What kind of functionality of the proteins behind it do we need to worry about, or we could think about for sensory, for health purposes? So these 5 themes underpins the main research question, which is alternative protein within our initiative. Now that's the end of my presentation pretty much. As I said, so I'll go back to the main slide. The website is there. There are a lot more information about our initiative on there. There are a lot more project information. It's not quite up to date yet, but I'm working on that. So with that, I'm happy to take any questions.
If there's a direct question to Dr. Minh Ha, you can put it in the chat if you like. For now, but we'll have a panel discussion later as well, where we discuss the rest of the topics. But yeah, okay. Dawson, would you like to introduce the next speaker?
Okay, if there's no direct questions for now, thanks for the introductions, Min. Now we'll move on to showcasing some of the all-pro research happening with the Future Food Initiative. First up with Dr. Gregory Martin. Dr. Gregory Martin is the theme leader of Proof of Concepts products with a Future Food Hallmark Research Initiative and an associate professor in the Department of Chemical Engineering at the University of Melbourne. His research in bioprocessing aims to develop mechanistic understanding of the behavior of biological materials and systems in relation to industrial processes. He heads the algal processing group that focuses on the development of efficient and scalable processes for the production of microalgal biomass and conversion into future food ingredients, fuels, and other bioproducts. The other main application area for this research is in food and dairy processing, which includes studying the properties and dynamics of proteins and lipids in food systems.
Thanks a lot, Dawson, and thanks a lot, Marion, for the introduction. It's a pleasure to be part of this event and great to see these student-led initiatives and on such an important topic as well. Yeah, I'll share my screen quickly. Let me know if it displays okay. Is that showing correctly?
Yep.
Yep, we can see it.
Terrific. Yeah. So what I thought I'd talk to you about today is just highlight some of the work we've been doing in terms of one form of alternative proteins, and that's the use of microorganisms, including microalgae. So I thought I'd start by just sort of outlining what some of the potential advantages and why we might be interested in these sources of proteins, and then try to give you a bit of an understanding of, I guess, what's unique and different about them and what's involved in terms of getting these as new proteins into food systems. And then I'll just give a very quick sort of snap highlight overview of some of the research that we're doing, but I'm not going to talk through the results in a lot of detail. I just want this to be more of an overview talk. So firstly, yes, I am a chemical engineer based in the chemical engineering department. So you might not sort of immediately think of chemical engineering and food, but I think particularly in this area of microbial proteins, there's a lot of work for chemical engineers to do. So starting off with, I guess, protein challenges, these really are on a global level, and presumably, you know, a lot of you in the audience at least know or aware of some of these challenges, and that's sort of partly why you're interested in it, because I think you know, they really are important and major challenges that need to be addressed. So I guess starting with the ultimate source of proteins, it really comes down to plants, and many of the proteins, as Min was talking about, that we consume are directly from plant crops. You might think of plant crops as being, you know, inherently quite environmentally friendly and sustainable, but there are still issues even involved in the production of plant crops. So if we think about, firstly, resource depletion, of course, to get the sort of yields that we need to feed the planet, we need to be adding fertilizer to our crops. And this represents potentially a resource depletion, in particular with phosphorus being a limiting non-renewable resource. And so this is something that is an increasing pressure. And if we think about the other key nutrient that plants need to grow productively, we need to add nitrogen. And in terms of the nitrogen supply for plant crop production, it's actually unfortunately largely derived from fossil fuel energy via the Haber-Bosch process, where nitrogen is fixed using a lot of natural gas. This sort of leads to the next major global problem, which is global warming, and of course the CO2 emissions associated with the combustion of natural gas to produce that nitrogen fertilizer. So the better we can utilize the nitrogen and the phosphorus, the more efficiently we can use the proteins that these plants are producing, the less resource depletion and the less global warming impact. So the next major area of pressure that the planet's facing is in terms of water, and particularly fresh water. And because a lot of the fertilizers are not added at the exact right amount, there's research being done to limit this, but there's always going to be some element of nitrogen and phosphorus runoff, which will contaminate waterways, and also the requirement just for water itself represents a pressure for irrigation. So as if that's not sort of challenging enough, unfortunately, as Min was saying, you know, there's a huge increase in the amount of animal proteins that are being consumed around the world. And when you look at the production of animal protein, it really is, in comparison to plants, extremely inefficient. So they're utilizing land less efficiently, and they're also, in terms of consuming plants, not a lot of that does get converted directly into protein that's consumed. So this puts even more pressure on land, which results in habitat destruction and soil degradation. And on top of that, animals, of course, like all creatures, will emit certain end products, including methane, which of course is a potent global warming greenhouse gas in and of itself. So this is where microbial protein production can potentially have some impact. And I guess if you think about microbes in a food sense, they've been used traditionally for a very long period in certain roles, in bread production, beer and wine and cheese production, but they're not yet at the level where they're making significant impacts in terms of displacing animal or plant crops. But if we could get to that level, basically the microbial production represents a much higher conversion rate of efficiency from materials that are ultimately plant derived, so things like sugars as an example, but they can also utilize waste from crop production. So this, in comparison to animal production, would represent a much more intensified system, which would therefore greatly reduce the amount of land that's required. And even in terms of, in comparison with plant crops, better utilization of these resources in terms of nitrogen and phosphorus. So going one step further, the other sort of aspect that we're looking at here is microalgae as a type of microorganism that's photosynthetic. So being photosynthetic, it can be grown ideally in an outdoor setting and basically substitute for a terrestrial plant-based crop. And because of the way these algae can be cultivated, they can be cultivated in such a way that all of the nitrogen and phosphorus are utilized. So this reduces the resource depletion, it reduces the global warming impact of nitrogen production, and it also will reduce nitrogen and phosphorus runoff. On top of this, algae can be grown in seawater, reducing freshwater demands, and they can be grown at much higher productivities, meaning that the land utilization pressures are reduced. So lots of reasons to potentially look at microbes in general and microalgae in particular. So for those of you who aren't familiar with microalgae, I'll just quickly sort of introduce you to what these organisms are. So fundamentally, they are photosynthetic organisms that aren't higher plants. So they're really, you can think of them as very simplified plants. And there are algae, which are macroscopic, things like seaweeds, and macroalgae, which are currently already used in some applications for food, whereas microalgae are single-celled organisms that are very small, but because of that can be grown in a very highly productive manner. So a key point about microalgae is that they're very diverse. They've evolved from a number of different branches in the phylogenetic tree, and this represents in some ways a huge bioresource that really is yet to be tapped into. So lots and lots of different types of algae, making lots of different proteins, lots of other components that we can potentially use in food systems. So in terms of, in general, what microalgae might be able to offer, of course, what we're talking about today is really the idea of protein, and they can be grown in such ways that they can have a very high protein content, even in the order of over 25%. On top of protein, though, they can also accumulate lipids and oils. They can produce carbohydrates that can have food functionality. They produce antioxidant pigments, and this is where we see most of the commercial application of microalgae at the moment for food applications is in the pigments. And then there's long-chain polyunsaturated fatty acids, EPA and DHA, which really only microalgae produce. Okay, so in terms of microalgal proteins, what are some key differences between these microalgal proteins and the proteins that they're trying to be an alternative to? And what opportunities, but also what challenges do these differences present? So the first point is, again, not only with microalgae, but of course, other microorganisms, there's a huge diversity of different species. And this diversity presents opportunities in terms of different proteins that can be produced, but also challenges in terms of trying to understand those different proteins and how they differ between different types of organisms. Another really key difference is the fact that most of the functional proteins that we use in food systems, so if you think of soy proteins or wheat proteins or dairy proteins, egg proteins, these are primarily, I guess, storage proteins that really, there's a limited selection and number of them, and they can be very well characterized and their behavior very well understood. If you think about microbial proteins, most of these are going to be intracellular proteins that are just accumulated as part of their natural growth system. And so there's going to be a lot more variety and a lot more diversity in the proteins that you're able to produce. They're also, the composition is going to vary a lot more depending on how the microbes are grown. So this presents an opportunity to control these systems, but also a need for understanding in terms of how to control those systems. In terms of the protein functionality, if you're going to take just a pool of intracellular proteins, they're typically going to have a pretty broad amino acid profile. That's good from a nutritional standpoint, but the functionality of these proteins is really yet to be characterized or explored. So this sort of represents a new frontier in some ways, with a lot of opportunity, a lot of resources to look into, but a lot of new understanding and knowledge that's yet to be developed. So that's sort of generally microbial proteins if you're looking at just growing cells and using the proteins they natively produce. But of course, there's the ability to tap into our biotechnological capabilities and use what's known as precision fermentation to genetically modify the microbes to potentially overexpress and directly produce food proteins. So this could be trying to ferment and produce milk proteins or egg proteins or other proteins that we want to substitute. So this is sort of a snapshot overview of some of the complexity in terms of microbial proteins and some of the things that still need to be determined. So if you think about the three sort of major flows of materials from producing microbes to using the actual proteins in a food application, you've got a whole bunch of variables that you can control and choose between in terms of your microbial protein production. So you can choose what type of microbe you want to grow, whether it be a microalgae, a yeast, a filamentous fungi. You can potentially choose to genetically modify it. And then you've got a whole bunch of choice in terms of how you grow these microbes. So in terms of algae, they can be grown outdoors, could be grown in indoor photobioreactors, classic fermentation type vessels, and then you've got a lot of control over the parameters. So all of these will influence ultimately what's your raw material for a microbial protein, which are whole cells that contain a pool of different proteins. And so it's what you then do with those proteins, which will determine how it functions in a food. And as I just mentioned, precision fermentation, the ability to make these microbes instead produce specific individual proteins that are of interest, is another aspect of microbial alternative proteins. So you can either use these individual targeted proteins, you can use the whole cells that have this pool of endogenous proteins, or you can potentially do some processing before you add these to food formulations. So this could involve rupturing the cells, breaking them apart to make the proteins more functional or more bioaccessible. You can then potentially recover the proteins as a purified product, or you could do an autolysis or hydrolysis type process to break those proteins up into smaller peptides that might be more digestible, they might be bioactive, or they might have particular flavor characteristics. And then you've got to consider how all of these different varieties of proteins will fit in terms of food applications. So understanding their functionality, such as flavor, gelation, solubility, and so on, what sort of formulations might they fit into? And you've also got to consider the non-protein components, given that these microbes will also be containing lipids, pigments, carbohydrates, and cell wall components. So hopefully that gives you sort of an idea of some of the complexity and some of the knowledge that needs to be established to really extract the true potential out of microbial proteins. And not surprisingly, in terms of the research fields, hopefully we've got people from different faculties around the university, and there's demand for knowledge and skills across many fields. So just from a technical standpoint, not even considering ethics and business and so on, from just a technical standpoint, we've got a need for industrial microbiology and biotechnology. We've got requirements in bioprocessing and chemical engineering, understanding the biochemistry, and of course, food science. So I'll just very quickly run through some of the research we've been doing in chemical engineering, and then I'm happy to hand over. So one of the things we've been working on is recovering food ingredients from microalgae. So taking our knowledge of chemical engineering, how do you scale this up to get food ingredients, including protein, from microorganisms, including algae? So we've been looking at things like energy efficient processing, understanding that you've got these single-celled organisms that need to be concentrated, they become a slurry. You need to understand how that material flows. You also need to be able to understand how to break those cells apart and potentially extract other components such as lipids. So we've done work looking at cell rupture in detail, so understanding how cells at a microscopic level can be broken apart in very energy efficient and scalable ways. We've looked at things like the biological factors that can influence how difficult it is to rupture a microorganism, and the difference can be orders of magnitude in terms of the amount of energy required to rupture different microbes. So looking at things like the cell structure, the cell walls, even going down to the point of measuring the physical properties of individual cells using things like atomic force microscopy. And that allows us to understand how we might be able to control and vary and select different organisms that will behave differently in processing. We've also done a bit of work looking at food emulsions and gels. So for example, using microalgae, once you've ruptured them, you've got proteins, but you've also got all these other cellular components. So understanding how they might interact in terms of emulsion formation or gel stability is really important from a food functionality perspective. An example of something we've done reasonably recently is taking microalgae and looking at how it might incorporate into a rennet gelation, so as a substitute or a augmentation of something like a yogurt or a cheese, and looking at how, whether you add whole cells or broken cells or proteins extracted from algae will interact in a cheese type matrix. And finally, some of the current work we've been looking at is trying to link things like the production of microbes to the actual processing, all the way through to peptides that might have particular characteristics such as flavors. So understanding how the cellular protein pool can be controlled by strain selection and controlled fermentation, how that can then be broken down using endogenous proteases as well as external enzymes to get flavor peptides that might have particular characteristics. So I realized I went through that pretty quickly, and hopefully I didn't take up too much time, but hopefully that gives you a bit of an idea of some of the work we're doing and some of the opportunities in this area of microbial proteins. So thanks again.
Thank you, Greg. Thank you so much, Greg. It was actually very interesting to learn that the protein challenge relates to so many other problems like global warming, resource depletion, water and land use. So it was really good that you've highlighted it, and how there's a role for microbes on microalgae to solving it. That was really highlighted. So thank you so much. So we actually have a question from the audience already about your research projects. So would plant protein solve most of the problems in terms of utilizing land and limiting CO2 production, or do we still need to make plant proteins efficient, they ask? I don't know if you can answer it, but do you have a question, answer for that?
Yeah, I mean, I think the substitution of animal proteins for plant proteins will have a major impact, and the more we can do that, the better. And then, yeah, if we can, on top of that, substitute some of the plant proteins or more of the animal proteins with microbial source proteins, or even microalgae is a more efficient crop. But I think it's going to be a combination of lots of different things, lots of different improvements that will hopefully get us to the point we need to get to.
Thank you. Another question, and this is for the supervisors. How do you see the research landscape in alternative proteins 5 years from now? So this is, I think it was a question from Min, but I'm happy for both of you to answer as well.
Greg, do you want to go first?
Oh, you go for a minute, that's fine.
Okay. So personally, I can't speak for everyone, but personally, I think the research landscape will be more dominated by alternative proteins. I'm not quite sure about 5 years, but certainly in the future. We've done, so, the bulk of research on mainstream proteins, conventional proteins, have been done in the last few decades. And yes, there's still a lot more innovation. We can do a lot more to reduce environmental impacts, increase yield, recycle more, and all of that. But we, at the same time, we also need research on other sources of protein, and really question ourselves, are there other ways to do this to achieve that same goal, but diversify our approach a bit more.
Thank you. Greg, did you want to add something, or should I go to the next one?
That's okay.
Okay, great. Okay, great. So we'll move on to the speakers now. Thank you for the answers. We're going to move on to the student panelists. We got Kieran Watkins. I'm going to give a little introduction. So Kieran is a PhD researcher at the University of Melbourne, working on ways to characterize conventional seafood to improve alternative proteins for the sake of the animals and the environment. In his spare time, he likes funny books, podcasts, and arguing about ethics. Welcome, Kieran. And I'll hand it over to you.
Thanks, Marion. All right, let me just share my screen. Okay. All right, can you see that?
Yep, we can see it.
Okay.
Might have to make it full screen.
Yeah. All right, does that look good?
Yeah, it looks great.
All right. So hi, everyone. So I'm just going to take you on a brief tour of my PhD research at Melbourne Uni. So I started it because I wanted to help the alternative protein field, and it turned out there was actually no publicly available database of seafood characteristics, which would be important to give companies benchmarks to create a product that met consumers' expectations. So some of the characteristics which are important are the color, which research shows is the most important sense people use when they're making a purchasing decision, because they believe with some validity that they can judge based on the color which products are fresh, which is ultimately what they're going for. And obviously, the cooked product is important to get right as well, because, for example, when you're cooking prawns, people use the color change as an indication of endpoint. The texture of the product is another central element to seafood, which consumers can try to judge before purchase, often by pressing their finger into the fillet to judge its firmness, which also correlates to its freshness. And of course, the cooked texture in our mouth is really important for satisfaction. The aroma and the taste of food of the seafood can really indicate the freshness as well. So many people who aren't fishermen don't know, but they actually smell like kind of lemony, grass cut kind of smell when they come right out of the ocean. But as they age, then they can release a chemical called trimethylamine, which is really pungent and disgusting. And there are other characteristics which I'm intending to study because they can help give these other characteristics. So for example, the protein functionality, which is basically just how the protein in the seafood reacts when you process it into something like a fish stick. So whether it holds water well, whether it can disperse fat to give the food like a rich mouthfeel, how soluble it is in water, and how it reacts to form a gel like fake crab sticks, which is called surimi or kamaboko in Japanese. And finally, the microstructure of the tissue can influence the texture, like the ratio of the collagen, the fat, and the muscle fibers, and also the size of the muscle fibers. So this all begs the question of how I was going to measure these characteristics. Well, I thought maybe that there might be industry or research standard methodology so that it wouldn't be so hard, but boy, was I disappointed. So starting with color, the people had found that the instruments they used changed the apparent color of the object, even though the object itself wasn't a different color. And to my bafflement, no consensus had actually been arrived at for a single methodology. And not only that, researchers noted that the apparent color didn't actually match what they were seeing with their eyes. And no one had seemed to develop a technique which accurately represented the color. For texture, there also hadn't been a standardization, even though many of the different factors when you're developing your method change the results. And how to measure the aroma, the taste, the protein functionality, and the cellular microstructure also hadn't really been standardized, which changed the results between researchers. So you couldn't really make a database unless you'd found the right method. So, thus my aims were to characterize existing methods. My aims were to test various existing methods to determine which one I would recommend the alternative protein industry use. And I'd base the recommendation based on how well the method correlated to how a human would determine that characteristic, how cheap and easy the test was to run, and how reproducible the results were. And then using these methods, my second aim was to just establish the benchmarks for a variety of seafood species on these characteristics. But to collect the data, I needed to actually just structure my experiments around answering questions so that you can publish papers, etc. So I'm looking at the effective display time on the characteristics, how the characteristics vary by location on the sample. So like, how does the color change over the fillet of salmon? and how the characteristics influence one another, like the effect of fiber size on texture. So starting with color, I think this is like really interesting. So on what basis could I decide that one technique was superior to another? I searched for a long time before stumbling upon a research group using a similar methodology to what I had designed, which is shown here on the right. So what they did basically is they tested a computer vision system, which is CVS in the middle, compared to a normal colorimeter, which is a machine designed just to measure color. And then they took a photo of the sample they were measuring and asked people to say which machine was able to represent the color better. So I'll just give you 10 seconds to look at all the animals, meat, and decide which one you think represented the color better. Marianne, do you want to hedge a bet?
CVS definitely looks better. CVS.
Yes. Yeah, by far and away, the camera data was able to create the swatches, which are these circles, which is more representative of the color than the colorimeter, which just blew my mind. So using this experimental paradigm, I'm trying to work out which measurement settings give the best similarity. So I'm varying the colorimeter. So I've got NIX, which is like a nice cheap colorimeter, a Minolta, which is kind of an old school machine, and a camera. I'm changing what is underneath the sample, so a black background or a white background, which might change it based on other research. I'm changing the thickness of the sample, which should interact with the background. And I'm finally working out how many measurements you need of the sample in order to get a good representation of the image, of the color rather. And I would have liked to alter the illuminant as well, which is basically just what amount of wavelengths of each light in the illuminating light on the sample when it's measuring the color. But there's no good way to do it. And then using these settings, then I'd be measuring the color and other characteristics for these species based on the fact that they're like quite high value and good targets for alternative protein companies to mimic. I'm also going to be, as I said, looking at the variation in the characteristics over the body location and also over storage time. So the data should allow me to draw conclusions about what either affects or predicts the other characteristics. And this is important because if there's a causal relationship, it would allow for research into how to alter the trajectories of production in order to improve the qualities. And if the relationship is just predictive, it allows for easy tests to estimate some difficult to measure characteristics. So here's just like a little web of what characteristics I expect to influence one another. As you can see, pH is quite influential because it alters postmortem metabolism, or rather, yeah, and can alter things like the protein functionality and the color, the microstructure, and how well it stays fresh. All right, so now I'm just going to give you a little sneak peek of the results so far. So for the color methodology development, so just a refresher, I put the prawns on 2 different colored tiles and then measured them with 3 different machines. And I also put them at different depths to see how that changed it. So looking at the top row, which is the 1 centimeter depth, you can see pretty clearly that the camera has this kind of brown color. It's capturing that orange, but also the white of the prawns, whereas the Minolta and NIX seem to have this kind of cream color, which I don't think is as representative. And as you may be able to see, there's not that much difference between the depths. But interestingly, for the camera, for the 1 centimeter depth on the black tile, despite how opaque I thought prawns are, it actually influenced the color quite a bit. So I thought that was just interesting if you had my priors. Moving on to the raw samples, you can see that as you stack up the raw prawns, there's quite a difference as a result of the thickness because the prawns are less opaque, and so it lets more light through to the ice behind it. And this had a greater impact when, for example, you've got a black tile because you can see that through the prawn. And as you can see, the NIX and Minolta, again, are just not really representing the color. They've kind of captured as this blue-gray kind of color, whereas the camera is really accurately perceiving that light brown. Yeah, and that's really all I have time for today, but I'd like to thank Min and Robin for being my supervisors and the Good Food Institute for helping inspire this project and maybe fund it. And happy to take any questions you have. Oh, and there are my references in case you don't believe what I'm saying.
Thank you, Kieran. I think we'll reserve the questions for the panel discussions.
Sure.
So I think we'll go to the next speaker for now.
All right, thanks so much for that, Kieran. It was interesting to hear about the wide array of benchmarks used in the characterization of seafood, as well as the different methodologies used today in this domain. And also personally, it's great to see someone from Effective Altruism being involved in alternative proteins research. So yeah, our last speaker for today is Brody Pease. Brody is a graduate of the University of Melbourne's Bachelor of Science program, where he majored in medicinal chemistry, inspired by the potential of cellular agriculture to revolutionize the world's food systems. He's now completing an honors degree undertaking research with the Future Food Initiative to hasten the advances of cultivated meat. So welcome, Brody. The floor is yours.
Thanks, Dawson. So you can see that it's all good?
Yeah, it looks great. We can see it.
All right. Awesome. So yeah, thanks, Marion. Thanks for the team for putting it on and inviting me to speak. And I guess I'll get started. So to start with, what actually is media? Well, if we take the Merriam-Webster dictionary definition, it's a nutrient system for the artificial cultivation of cells or organisms. So a nutrient system. What nutrients exactly? Well, unlike koalas that eat eucalyptus leaves, or cows that feed on hay or pellets, or even humans that eat hot dogs or burgers or pasta, cells don't have a complex digestive system with a mouth to break down physical structures or a stomach to enzymatically decompose complex molecules. Cells need their food in the rawest form, in liquid form, composed of inorganic salts, which will provide them with all the minerals that they need, amino acids, which are the building blocks of proteins, vitamins, and growth factors. Growth factors are a complex type of protein that signals to the cell it's time to grow, whether it's time to divide or it's time to mature, and fatty acids. Cells need the fatty acids to form their bilipid membrane to encase themselves. These are all essential components of cell media. Some media also contains sugar, like glucose, to give cells that extra bit of energy they need to grow. And media may also contain an indicator, which might be phenol red, which is red when the nutrients are at a high concentration. And then as the cells consume the nutrients lower in concentration, changing the pH, and then changing the color from red to yellow. So we know that it's time to change the media. The most important part of media is the growth factors. Growth factors are complex and hard to come by. And the most common way to get them in culture media is through FBS. FBS is fetal bovine serum. So what exactly does that mean? Fetal is from the unborn fetus, bovine meaning from a cow, and serum meaning blood without cells, platelets, and clotting factors. So this FBS is the major source of growth factors in cell culture. But that doesn't come without complications. FBS is a byproduct of the animal slaughter industry. When a pregnant cow is taken to slaughter and cut open to find a fetus inside, that fetus is then taken aside, injected into the heart to extract the blood to get the serum. And because a fetus is alive at this point in time, it's expected to feel this pain and to experience that during the process and afterwards. Around 500,000 litres of raw FBS is harvested every year. Now, that might be adequate for the biomedical industry, where the production volumes are quite small. But when we're talking about cultivated meat as a food product, 500,000 litres is not nearly enough to supply the large volumes that companies are trying to produce. And batch-to-batch variability. Because we're obtaining this FBS from animals, depending on the stage of gestation, whether that fetus is 3 months old or 6 months old or 9 months old, that's going to change the concentration of the different components of the serum, change the growth factors or the lipids or whatever else there might be in there. And this causes inconsistency. And this is undesirable in science and can affect the outcome of our results. And finally, FBS is expensive. So what is the solution? On scientific, moral, and financial grounds, the obsolescence of serum use in media is required. We need to develop serum-free media. So many serum-free media formulations actually currently exist. And many cultivated meat companies have produced their own media or are in the process of producing their own media. Companies even exist that are specially just making media, such as Maltus Media or Australia's own Huros. However, the problem is it's still expensive. Growth factors don't come cheap, at least not yet. Which leads me to my research project. So after I identified that culture media was a key component to the success of cultivated meat, I met with Min and Robin, my potential supervisors at the time, to discuss the prospect of an honours project. I then spent the Christmas holidays researching and preparing a literature review, and what I found was 2 key things. The first being nitric oxide initiates myogenic differentiation. Nitric oxide is a small but potent molecule that consists only of a nitrogen atom and an oxygen atom. And it initiates myogenic differentiation, meaning it initiates the signaling to the cells that it's time to stop multiplying and it's time to fuse together to form the muscle tubes that make up meat. And the second thing that I found was calcium regulates nitric oxide production. Calcium regulates the enzyme nitric oxide synthase that creates nitric oxide. But what I didn't find in the research was any evidence of using these components in combination. Could we use calcium and nitric oxide together to increase myogenic differentiation? Which led me to ask my question, my research project topic, accelerating C2, C12 myogenic differentiation using nitric oxide precursors and calcium. C2, C12 means, it's the name of a type of cell, a mouse skeletal muscle cell. And we're using nitric oxide precursors. Because nitric oxide is so quickly broken down in a biological system, we need a precursor that will store the nitric oxide until it's released by an enzyme when it's needed. So one precursor we could use is arginine. You've probably heard of it before. It's a semi-essential amino acid in our body. Another one would be citrulline, another amino acid, although less well known. So how the pathway of these 2 precursors work. So nitric oxide synthase, or NOS for short, takes arginine and releases nitric oxide. And the byproduct of this reaction is L-citrulline. Citrulline can then be recycled back to arginine and used as a precursor. So we can choose to use either arginine or citrulline to provide the nitric oxide needed by the cell. This begs the question, which is better, arginine or citrulline? At what stages is it beneficial? Is it beneficial only at the start of myogenic differentiation, or is it beneficial for several days? And does adding calcium enhance the effect? Unfortunately, I don't yet have the answers to these questions, but I do have some research and some results to share with you so far. In my first experiment, I just wanted to determine simply if using these nitric oxide precursors without serum, was that enough to keep the cells alive? Serum, remember, contains the growth factors needed to signal to the cell to grow. And the results were quite clear. This is not a picture of the moon. This is actually what wells 7, 8, and 9 look like under a microscope. And this is what wells 10, 11, and 12 look like under a microscope. Can you guess the difference in the media conditions? In the first one, wells 7, 8, and 9, the media contained FBS, the growth factors in the serum. Those cells grew and they stayed alive and they've stayed attached to the surface of that cell plate. And that's why the media has turned yellow, because the nutrients have been used up and so the pH has changed, and now I know that it's time to add new media. However, in wells 10, 11, and 12, I didn't use FBS. And because there was no growth factors, the cells weren't able to survive and they've detached from the surface because they've died. And that's why the media is still red, because the nutrients have been left untouched. So from this first experiment, I learned that nitric oxide precursors alone are not enough to sustain cell growth. In my second experiment, I wanted to get a closer look at cells. So I got in the lab and I created some stains. The first one was hematoxylin, and the second one was eosin. I thought I would save time on staining these cells by using the whole culture plate to stain, rather than individually putting each cell sample onto a glass slide. The problem with this is that I didn't realize xylene was a solvent of polystyrene. And so when I put my plastic polystyrene plate into the solvent, it quite literally began to dissolve the plate and melted away the cells and any results that I had. So this experiment didn't go very well. During this experiment, I also collected the waste media because I wanted to do an assay and test the concentrations of the nitric oxide metabolites, which would give me an idea of how much nitric oxide was used, but all the readings came out too low. So no results there either, which led me to experiment 3. We tested not only media with different nitric oxide precursors, but also different volumes. We looked at 0.25 mils per well, 0.5 mils per well, and 0.75 mils per well. Now, looking at the 3 charts, the 3 columns on the left, we can see that arginine at day 1 clearly had the highest concentration of nitrite, meaning more nitric oxide was used during the growth of the cells than media alone. Then from the other samples, we can see on day 1, 0.25 mils had the highest concentration, on day 3 the same, and day 5 the same. So what we learned was that yes, adding arginine and nitric oxide precursor meant more nitric oxide was used in the growth of the cells. And we also learned that we could use as little volume as 0.25 mils to successfully grow the cells. With new cell samples came a new opportunity for staining, which went much better than last time. As you can see in this picture, the hematoxylin is used to stain the nuclei of the cells, which are the small purple dots that you can see. And the eosin stain the structural components of the cells, which are the long purple structures. They are the myotubes that bind together to form the muscle fibers that we know in meat. From this image, we can calculate the ratio of nuclei outside the myotubes to nuclei inside the myotubes. This ratio is called the fusion index, and we can compare the fusion index between different culture samples to compare the media with arginine, without arginine, with citrulline, without either. And from that, we can determine which media conditions accelerate myogenic differentiation the most. So this pretty much brings you up to speed on where I'm at with the project so far. I hope you learned something, and that's about it. Thank you.
Thank you, Brody. It was really interesting because I know that FBS has been seen as essential pretty much when growing cells. So it's awesome to see that there's serum-free media being developed, and that you're doing more research on it to make it more accessible. And I guess that's really important for cellular agriculture, for it to take off and scaling up and actually going on to make impact as well. So really awesome.
Thank you.
All right, great. Thank you, everyone. Thank you, everyone, for the wonderful talks and giving insight into some of their awesome research projects that they're working on. So yeah, I think next up, we'll go on to the panel discussion. So to the participants, add in any last minute questions you have on Slido, and we will be answering them. I'm gonna kickstart the panel discussion. First, this is a question for everyone. What made you decide to research alternative proteins?
Who wants to go first? I guess I will.
Go on, Kieran. Yeah.
All right. Basically, I'd been looking for ways to reduce animal suffering in farms and slaughterhouses. And I was listening to a podcast with a guy called Bruce Friedrich on it, who used to be the leader of PETA, but now led an organization called the Good Food Institute, who promote alternative protein research. And he basically made the case that throughout like the last 100 years of advocacy for like changing people's diets, we'd shifted 0% of people towards a more vegetarian diet. And so what we really needed was better alternatives so that people would choose it on their own. So I thought, rather than try to keep trying to convince people, just give them something which they prefer.
Okay, I'll go next.
Yeah, go for it, Brody.
Yeah, I got into it because I finished my degree in medicinal chemistry, and that was really my passion at the time. But I realized if I wanted to pursue a career designing drugs and testing drugs, that would inevitably mean animal testing and constantly anesthetizing animals, doing things like that. I didn't want to do that. And then my dad actually suggested, why don't I look into cultivated meat? Because whilst plant-based food is great, people are never going to be satisfied until they're getting the meat that they know and love. So how can we produce that in a way that doesn't have the environmentally and ethically damaging parts to it? And that's how I got into it.
Awesome. And Greg as well, if you'd like your 2 cents.
Yeah, sure. Yeah, just quickly, I mean, I guess it's about, trying to apply skills and capabilities to something that you believe in and you think will make a difference. And for me, I think that's an area that's true for. And I guess I started off with bioprocessing, looking at renewable energy and biofuels, but the time horizon for that is, pretty far. I think, the commercial aspects just aren't necessarily there, whereas this is, I think, a new horizon with sort of a good balance between something that can be scalable enough to have a good environmental impact, but also you're dealing with a high enough value product so that it can sort of be commercial reality in a reasonable time frame.
Awesome, thank you. I'm getting questions from the participants as well. There's a question for Brody. What cells are you testing the nutrients on? Someone says.
Yeah, so they're C2, C12 cells, which is a very well-established and immortalized cell line that's been around for decades. It's a skeletal muscle cell line, and yeah, you can replicate them and passage them tens and tens of times, and they're still consistent.
Awesome. This is a question for, I think, Greg. I'm planning on doing a research subject in my undergraduate degree. Is this research area of alternative protein available to participate in? Are there research opportunities there?
Yeah, I mean, it depends what faculty you're in, but I'm sure there would be. And the Future Food Hallmark Initiative is probably a good place to look for supervisors in your faculty. But yeah, there's definitely some in engineering and FAS and probably other parts as well. So yeah, a lot of the university is involved in some way or another. So yeah.
Awesome. Riley, I see you have a question. Go ahead.
Thank you. Sorry, my question was about the cells Brody was testing on, and I have a follow-up question on that. about, I'm not sure, I'm not an expert in any of this, but my sister's doing something with differentiation media, and she's interested in how that's an ethical way of getting ethically differentiation media. Do you know anything about that? And what differentiation media you guys use?
Yeah, so that's what my project is looking at. So we use DMEM, which is Dilbeco's modified eagle medium, with 2% horse serum, and then just Penstrep, which is a type of antibiotic and antibacterial and antimycotic. And what I'm doing is trying to use nitric oxide precursors and calcium unsuccessfully to replace FBS, but now we're just trying to use it as an additive to see if we can accelerate the differentiation.
All right, thank you.
Awesome. Baron has a question. He asks, very interesting line of work on seafood characterization. Which all species will you be investigating? Baron says hi, Kieran.
Hi, everyone. I'm planning to study Atlantic salmon, scallops, Western rock lobster, and abalone. Abalone is interesting in the sense that it's got its muscle is smooth, the same way our intestines have got smooth muscle rather than the striated muscle of our skeletal system. So that should be interesting.
That is really cool. And this is a question for everyone. Will your research be an open source and available for the wider ecosystem?
Mine will be, yes.
Awesome. Rody, is that true for you as well?
Yeah, so my grant company, New Harvest, one of the requirements is that you publish open access.
Awesome. And is that quite true for most of Future Food Initiative as well, Greg?
Yeah, most of the research that's done is published in journals. But yeah, we also work with industry, and occasionally there'll be some confidential things that remain in-house, but yeah, most of it's published.
That's awesome. Okay. So Roddy and Kieran, how did you go about finding a research project with the Future Food Initiative? That's from a perspective of, we want to do research, but we don't know how to go about it. Do you have any tips or your experience on finding your project?
I think I got kind of lucky in this regard. So I'd been looking at the Good Food Institute's like problem space and found this project basically. And then just like looked online, and it turned out there was a researcher at Melbourne Uni where I was living already who could like help me with this project. And so I just started like that, just emailing her, asking whether she'd be interested in working with me.
Yeah, pretty lucky. Yeah.
Yeah, but in general, people, you should check out the Good Food Institute's ASAP page, which I'll link in the chat.
And also Future Food supervisors. If you go on their website and just checking the supervisors, see if the research is aligned with what you want to do, and then go for it. Rody, do you want to have an input? Do you want to put your 2 cents?
Yeah, I had no idea. I just Googled Cellular Agriculture Australia, and that happened to be the name of a non-for-profit in Australia. And then so I contacted the director, Bianca, and she put me in contact with Min. And then I did the literature review, like I said, and then everything worked out perfectly. So I consider myself very lucky as well.
Yeah, so I guess these connections that you make in the fields that you're interested in, very, very useful for research projects.
Yeah, I definitely say network. Get to know as many people as you can.
Nice. Okay, what about if you're interested in a specific? Okay, this is for Greg, I think. What about if we are interested in a specific part of alternative protein already? Can we create our own research projects? And how do we go about doing that?
Yeah, definitely. So there are often projects that supervisors will have that are reasonably predefined, and they're just sort of looking for a student. But most supervisors are very open and happy to have students sort of knock on their door or send an email with an idea that they're interested in. And it might be that it ends up being tailored or tweaked depending on the capabilities of the supervisor, and it might not necessarily go exactly in the direction that you think, but yeah, definitely a good starting point for a conversation, and supervisors like seeing that. So that can be true for both, master's level research projects, but also for PhDs as well.
I see. So alternative protein is a very broad field with so many different applications, things to research. So it's really awesome to know that we can pretty much follow up most things, yeah, and just go from there and find a research project that works for you. All right, do you have tips if we want to research alternative proteins but aren't sure exactly what specific area in AltPro? Do you have any tips or advice for finding a suitable project? I think this is for anyone. But Greg.
Yeah.
Oh yeah.
Oh, sorry.
No, Kieran, you go.
The organization Brody was talking about, Cellular Agriculture Australia, they built this tool to help people even before they get to uni in order to figure out like what they could research. And if you've already got your major planned out, you can pop that into their tool and it'll tell you what problems you could potentially solve based on that. So I'll chuck a link to that in. Just have a wander in the tool. It's pretty interesting to see all the things that you could do.
Yeah, awesome. So I think this is like a general question on the net. We want more people to go into alternative protein for all these reasons of saving the world. What do you guys think would be necessary for AltPro so that there's more talent going into this field? Like, what do we need to do more of? Do you think? I know this is a very tough question, isn't it? Just put you guys on the spot.
Yeah, I don't really have a good sense of the whole field in terms of what is most in need. I think given the scope of the problem, if you've got interest, then you could probably contribute. So don't be shy.
Yeah, don't be shy. Yeah, and also as I can't remember who said it, but definitely check out the white spaces in alternative protein. I think GFI, Good Food Institute, has a good list of that. So that might be a good place to start if you're interested in contributing. Yeah, how much funding is there in alternative protein research? What does it look like for the Future Food Hallmark Research Initiative?
Well, just recently, I don't even know if they've announced it publicly yet. I'm not sure if I should say this, but I'll say it anyway. New Harvest, who is my grant company, won't be doing grants next year.
Oh, okay.
So I think in that regard, the only company that I know of that would be doing cultivated meat research grants would be GFI.
I see. Okay. Kieran, Greg, did you have inputs on that as well?
Yeah, I mean, I think it's something that's gaining momentum, and there are sort of established large multinational food companies that are investing in this space as well. So, there are opportunities, and I think they'll continue to increase over time. And in the meantime, there is a lot of fundamental research that needs to be done. So I know a lot of labs and universities are seeking funding from things like the Australian Research Council and getting these fundamental grants, as well as directly from industry.
Kieran, did you have something as well, or? No? Okay, cool. Another question, what are the most significant barriers that academics, researchers in alternative proteins face today? Do you have an input on that, Greg?
Yeah, I mean, I don't necessarily see there being barriers to research. I mean, getting research funding is always a bit of a barrier, but I think, there's lots of exciting research questions to formulate here. So going for that fundamental funding through avenues like the ARC is definitely valid in this space. And I think we're lucky in this area to have organisations like Cellular Agriculture Australia and GFI, which have been mentioned a few times, that have really helped sort of pull people together and focus the field. So I think it's a bright future in this space. Yeah,
I'm really excited all the advancements made in this field. I think there's another question. Could you describe, if any, the relationship collaboration between academics and regulators in the approval of commercial sale of cellular agriculture products? Is there an interaction between the academics and regulators for these products to go to market and their applications in the market?
That's really not my area, but
yeah. Oh yeah, that's true. I mean,
it really is an important focus. Yeah.
Yeah.
It's not. my area.
Yeah, and I feel like that is a very important question. And for people who are thinking about doing a research project, that might be a good research topic as well. Like we definitely need these kind of research done. So anyways, I think we've used up all our time. So I think we will conclude there. But before we conclude, do you guys have anything you'd like to say?
Just in regards to the regulation question, I don't know anything about it, but there was a talk done by Dr. Hope Johnson in partnership with Robin Warner, and she talks about agricultural regulation. So if you wanted to know more about that, you could look that up. I can have a quick look for the link.
Okay.
It's definitely a topic that's covered as part of the Hallmark initiative.
Mm.
So yeah, there are
It's
people
not
in the
covered?
university thinking about it.
Yeah, I think because there's so much good research going on, but if, we need to be able to apply that in the market to make impacts. So definitely it's an important area, I think. Okay, yeah, Kieran and Greg, did you have anything to add before we conclude the session?
No, I do not. I didn't have pearls of wisdom to share at this last moment. Sorry.
No, just thanks again for organising this event. That was really good.
Indeed, that was fresh on my mind as well. Thank you, Greg.
All right. Oh, thank you so much for the speakers today. It was really awesome listening to all your research projects or research topics. I'm also considering alternative protein research in the future. So this has been very useful for me. So thank you all. So anyways, this has been a wonderful time. Thank you all so much for coming. And I hope that it was an interesting time for everyone and that you're also excited about the developments in alternative protein and that you have some like a better idea of how to get involved in research if you're interested. So thank you, Greg. Min is not here, but thanks to him, Kieran and Brody. And thank you all so much for coming and have a wonderful rest of the day. Until next time.